Hello and welcome to my presentation. My name is Shahin. I'm a last year PhD student working at the Multimodal Imaging and Connectome Analysis Lab at the Montreal Neurological Institute. My work focuses on relational memory in humans, both in health and in disease. Relational memory consists of cognitive processes that bind the various elements of subjective experience. These include episodic memory, in which autobiographical events can coalesce into distinct episodes, semantic memory, where concepts can merge into general categories of meaning, and spatial memory, where the location of objects in space in relation to other objects can converge to form what is typically known as a cognitive map of the environment. Modern neuroscience lacks a standardized and holistic framework for the multidimensional assessment of these different types of relational memory. And in the age of big data and multimodal imaging and multivariate statistical analyses, I just had to do something about that. And so was born the Integrated Relational Evaluation Paradigm, or simply iREP. In this presentation, I will discuss how we leverage the iREP in conjunction with complementary multivariate statistical approaches to examine the behavioral difference in relational memory between healthy individuals and patients diagnosed with temporal lobe epilepsy. We examined 20 TLE patients and 53 age and sex-matched controls. All participants underwent multimodal MRI neuroimaging, including structural sequences, in addition to task-based acquisitions during which the iREP was administered. The iREP is a user-friendly Python-based program composed of 3 complementary modules: the episodic task, the semantic task, and the spatial task. The episodic task is administered in 2 separate runs, one encoding and one retrieval. During the encoding phase, pairs of images are shown at each trial. Half are displayed twice and the other half only once, as you can see. Thus, duplicate trials account for the easy condition, since the second exposure facilitates memory encoding, and single trials account for the difficult condition. During the retrieval phase of the episodic task, an object will appear at the top of the screen and 3 objects will be displayed at the bottom. It is the job of the participant to select the item that was originally paired with the object at the top. Unlike the episodic task, the semantic module is administered in a single run and does not have an encoding phase. Once again, an object will appear at the top of the screen with 3 options below. The participant must now choose which option is conceptually most alike to the top item. Here too, we have an easy condition in which the correct response is fairly obvious, and a difficult condition in which it is a little less so. The spatial module also comprises distinct encoding and retrieval phases. But unlike the episodic, the spatial task is administered in a single run. At each trial, the participant must first encode the spatial arrangement of 3 objects. Within the same trial, the participant will have to select the same encoded configuration among 3 options. In the easy trial, the geometry of the correct spatial arrangement differs substantially from the other options, whereas this difference is minimal in difficult trials. We calculated percent accuracy scores for each condition in each task. Here the results are displayed for both cohorts, healthy controls on the left, TLE patients on the right. Red box plots correspond to the difficult condition in each task, and yellow box plots correspond to easy. We used linear mixed effects models as a starting point, with task accuracy as the outcome measure. Our model consisted of a group factor with 2 levels, healthy control and TLE, a task factor with 3 levels, semantic, episodic, and spatial, a condition factor with two levels, difficult and easy, interaction terms for task and condition, and also group and task, and a single random subject term. We decomposed significant interactions and, where appropriate, ran post hoc pairwise comparison tests while controlling for the false discovery rate. To provide additional context for the behavioral differences across groups, we implemented partial least squares correlation to determine if performance on the iREP was associated with specific demographic and clinical phenotypes. These features were age, sex, presence of TLE diagnosis, as well as left and right hippocampal volumes. Following linear mixed effects analyses, we were able to ascertain systematic differences in how participants performed as a function of task condition. As you can appreciate, accuracies were significantly higher on the easy condition relative to difficult, regardless of the task. Furthermore, semantic scores were much higher than either spatial or episodic, and this was true for both conditions. On the easy condition only, overall performance was better on the spatial task relative to episodic. Healthy controls outperformed TLE patients only on the episodic module, and while both groups scored highest on the semantic task, whereas healthy controls performed poorest on the spatial task, TLE patients found episodic to be most challenging. We observed that the first latent variable obtained via the decomposition of the cross-correlation matrix between clinical phenotypes and iREP accuracies accounted for a whopping 85% of the total covariance, and the correlation between latent scores along this first component was also evidently quite notable. To test for the significance of the first eigenvalue, we ran 5,000 permutations resulting in the present null distribution, against which the original singular value, which is 0.82, was compared. As you can see, the result was highly significant. We also applied a bootstrapping procedure with 5,000 iterations to assess the reliability of clinical and iREP loadings on the first component. This slide is very telling in that it shows that younger age, the absence of TLE diagnosis, and larger left and right hippocampal volumes collectively make up a phenotypic profile that, irrespective of sex, is associated with better multidimensional relational memory performance. In this work, we wanted to show that it is possible to examine the different dimensions of relational memory all at once and across different population cohorts. And we did this by leveraging a novel and holistic framework known as the Integrated Relational Evaluation Paradigm, or simply iREP. We use the iREP in conjunction with multivariate and data-driven statistical approaches to evaluate the differences in behavioral performance across healthy controls and patients diagnosed with temporal lobe epilepsy, or TLE. We found that performances were highest on the semantic module relative to the other two tasks. The main difference between groups was seen only in the episodic task. And the healthy controls clearly outperformed TLE patients in that module. We were also expecting to see similar findings in the spatial task, but given the relatively small sample size of the TLE cohort, examining larger groups may yet substantiate this expectation. Moreover, we also found that being relatively young and healthy and having relatively large bilateral hippocampi were also indicative of better behavioral scores across all the iREP outcome measures. I hope you enjoyed this presentation, and Ask me anything.